Hi, this is Professor Tiffany Martin with Medical Microbiology, and we are in Chapter 7, Microbial Genetics, and we are at the at the point of DNA replication, and we are looking at it at the molecular level. What is going on inside the cell? How do these proteins and enzymes and molecules and nucleotides all interact to get a copy, an accurate copy of DNA? So that's what we're going to look at next, okay? So let's go to the PowerPoint. So how does replication start? It starts, everything has to have a start point, and what we call the starting point of DNA replication, we refer to that as the origin. So on your chromosome, there is an origin, and that's where DNA replication starts. Now, in a prokaryotic circular chromosome, there's actually only one origin. I kind of drew a little circle here from that. So there's my circular haploid bacterial chromosome, and there is a place that we refer to as the origin, and there's only one for a bacteria cell. Eukaryotic cells, a lot more DNA in there, and linear chromosomes, they will have multiple origins. And so DNA replication, the process that we're going to start to talk about, starts at each one of those origins. But in the bacteria cells, it only happens at one origin. Now, when I refer to origin, remember this is nucleic acid. It's a circle of nucleotides put together, and so it's nucleic acid. So when I'm talking about an origin, I'm talking about a series of nucleotides in a specific order that spell out origin. Start DNA replication here. G, G, A, C, C, T, T, G, G, C, D, G, whatever that sequence is, it spells origin, start here. And how do you know that? If I give you the letters S-T-A-R-T, you can put those together and read it and say start. That's what we mean. There's a series of nucleotides that when you put those together in a specific order, it means start here. It's where the enzymes that we're going to talk about are going to bind and start here. All right, so that's the origin. Back to your PowerPoint, one origin for prokaryotics. The next thing that has to happen is a process called strand separation. I think I'm going to go back to my whiteboard, and then we'll come back and fill that strand separation in. So here is my shorthand version of my double helix, and we can see that the first thing that needs to happen, and you saw that in the template concept, is we need to separate these two strands. How does that happen? So I've got my nucleotides labeled. I've got 5' 3' labeled correctly. How does this happen? Okay, the enzyme that starts this process is called helicase. And helicase is an enzyme that's going to find the origin, and it's going to bind, and it's going to start separating the strands. So let me write the name of that in. So the name of this enzyme is called helicase. We know it's an enzyme, ACE, right? Helicase binds into the DNA and separates these strands. And it does that by breaking hydrogen bonds. So it's going to break one hydrogen bond at a time, and it's going to continue breaking hydrogen bonds until these strands are separated. Okay. A lot of times you'll hear that referred to as unzipping, because it kind of does look like a zipper, right? And you're unzipping this, and it's by breaking these individual hydrogen bonds, which are easy to break one at a time, and then these two strands are now separate. All right, helicase, so that happens first. Now, once I've got these two strands separate, maybe instead of thinking of it as a zipper, think about Velcro. If these were two strands of Velcro and they were getting separated, and I had very long strands, you can see how these two strands over here are going to want to come back together, right? They're sticky, they make sense, they bind. So we're going to need a way to hold these strands apart. So the next thing that comes in are these proteins called single-stranding binding proteins. Single-stranded binding proteins do exactly what they say. They bind to single-strand DNA. So SSBP, single-stranded binding proteins, come in. Every time they see single-stranded DNA, they bind to it. That's just what they do. So they're going to do their job over and over again. Helicase is unzipping, creating these single strands. These single-stranded proteins, single-stranded binding proteins come in and they bind, and that just keeps it from going back together until we can get the replication, other enzymes involved. Okay, so that's involved in strand separation. Now there's one other thing I need to do, another enzyme that I need to tell you about that happens, and this is an enzyme called a topoisomerase. And now when we think about this, now I've kind of drawn this kind of straight across, but remember this is a double helix. It's a circular corkscrew. Can you think of like a phone? You remember those phones that attach to the wall? I know you got to go way back, but you know that kind of circular corkscrews that cords have from there? That's what's going on here. But as these strands are pulled apart, they actually put some pressure on this corkscrew and it ends up supercoiling and kind of so I've got my nice double helix, but then as the double helix pulls apart, this ends up curling and curling around itself until it makes like a super coil. So if you can imagine a telephone coil, it's got that coil in it, but then if you twist it, it makes a super coil. And anybody who knows some physics and how cords work, you're putting some pressure on that, and these are just chemical bonds. They are susceptible of breaking. Do you want your instruction manual breaking apart and being pieced back together? No, of course not. So we need an enzyme to protect this part of the DNA from the pressure that is coming by pulling these strands apart. So this enzyme is called topoisomerase. You can say that one. That one's a hard one. Topoisomerase. And this is an enzyme, an isomerase is a type of enzyme that just rearranges stuff. It doesn't add anything or take anything away, it just rearranges itself. And this is called topoisomerase for that. So that's what's happening here. And the way it does that is this enzyme binds to double-stranded DNA. And remember, it's got a lot of pressure on here. And it actually nicks that sugar phosphate backbone. And that allows it to uncoil. And then after it uncoils and that pressure is relieved, it's able to put it back together and put that back together. So that's what topoisomerase does. And it actually moves along the chromosome in front of helicase. So as pressure is building, it'll bind, release that pressure, and then move forward. So these enzymes work together to make sure that your DNA doesn't break as strand separation has occurred. Okay, so this is strand separation. Let's go back to our summary slide and see how you're doing. Strand separation. So you take a chance and go through here and go through these enzymes and see if you can figure out what's doing it. All right, first one. What was the first one? Helicase. What's its job? Unzips the hydrogen bonds. Second step that comes in for strand separation. Single-stranded binding proteins. What's its job? It binds to single-stranded DNA and it keeps it separating during that unzipping process. A third thing that must happen in strand separation, topoisomerase, an enzyme that keeps strands from becoming tangled by releasing that tension. It breaks that sugar phosphate bond. Okay. All right. So we're going to stop that from there. This is the first stage of DNA replication, and this is referred to as strand separation. So we'll do the next video where we'll talk about strand elongation. Now that I have it separated, how do I actually make the new DNA from that? So I'll see you in the next video.